All right, so this morning, welcome to Micro Seminar for, no, it's still September. September 2016, almost to October. That caught me up there. Today's seminar speaker would be Dr. Paula Welander. Is that Welander?
Yes, yeah.
Welander from Stanford University. Paula's been at Stanford since 2013. Before that, she was a postdoc at MIT with a couple people. So she started with Diane Newman and then went to Roger Summons' lab. After that, she was a postdoc for about 5 years there. And before that, she was a PhD with Bill Metcalf at Illinois. And before that, she was actually an undergraduate in Mark Martin's lab. A lot of us know Mark Martin. So she was an undergraduate in Mark Martin's lab at Occidental College in LA. So without further ado, I hand it off to Paula.
Great. Thank you, David. And thanks to you and Jen and Cameron for having me. This is great. I've never done this before, so we'll see how it goes. But I'm really excited to share the work that has been coming out of my lab the last few years. Particularly, we've been focusing on sterile synthesis. And I like to always start my talks with this slide that I got from Brock's Microbiology, which is a textbook I use for my micro class. And I love this slide because it really kind of gives us a perspective of the evolution of life over time. And I think a lot of times with my students, they realize the role that microbes play in our environment now and how important they are, but they don't have the context of geology and this context of geologic time and the important role that microbes have played over many years. And so you can just see here that it indicates in yellow that the origin of life occurred about 3.8 billion years ago, and we have different types of organisms appearing over time. And what I really emphasize is that for 3 billion of these years, all life on this planet was microbial. And so microbial life is really important, not just now, but in the past as well. And as geobiologists, we're really interested in trying to understand this interplay of coevolution of life and Earth, how life affected the Earth, and in turn, how the Earth affected the evolution of life. And so as geobiologists, if we really want to study the Earth in the past, and this interplay, we need to understand microbial life in the past as well. And so that leads to a challenge that we have in the field of geobiology, and is that how do we study microbes in the context of Earth's distant past? Geobiologists have come up with a lot of really cool strategies to try to actually link microbes to the rock record and link them to ancient events. Things like microfossils, primarily you can see stromatolites billions of years back. There are different types of signatures and properties in rocks that can be attributed to microbial life. And these can be controversial, they can be difficult to interpret, but still I think they're a powerful way of trying to actually link a specific microbe to the past. Another way of looking at ancient microbial life is to look at stable isotope signatures. So microbes are, something that came out of geochemistry that's really cool is the fractionation of isotopes, and that microbes actually, and a lot of different types of life, can fractionate carbon and other elements, and these can become signatures, and there are patterns to this fractionation, and geochemists can go in and detect these in rocks that are certain ages and then link certain metabolisms to that time as well. And today what I want to talk about is molecular biosignatures. And these are actually molecules that are produced by microorganisms and preserved in the rock record. And they can be used as fossils to kind of indicate microbial life in ancient metabolisms. And how that works is actually quite straightforward. You have some ancient microbe in an ancient environment producing these types of lipids. The microbes die and settle into the sediment. And in the process of that sediment becoming a rock, this process of diagenesis, the majority of the organism is lost. DNA, RNA, that stuff isn't preserved. But things like these lipids are actually well preserved. We lose some of the hydroxylations that you see on these different molecules, but the basic structure can be maintained. Geochemists are able to go into these rocks and actually extract them and detect them. And they have methods of looking at these molecules and making sure that they're indigenous to the rock and they're as old as the rock is itself. And then they can make interpretations based on what organisms today are actually producing these molecules. They can look at different types of phylogenetic groups that may have been present, their metabolisms that were there, and then they can even look in modern environments for these lipids and link these modern environments to ancient conditions. Okay, and so where we come in this aspect of biomarker work, where we want to really help to bolster interpretations and really help make interpretations more robust. And so how do we do that? We're a molecular microbiology lab, so we focus on looking at these molecules in cultured organisms. And what typically we've been focusing on is actually trying to identify the genes that are required for the biosynthesis. And we typically do this by going into a genome and doing some bioinformatics and looking for potential candidates. I find these potential biosynthesis gene candidates, and then we can make knockouts. And when we make knockouts, we verify that these proteins are actually involved in the biosynthesis of these molecules. Once we do that, that opens up a whole wealth of experiments that we can do. One, we can look in genomic and metagenomic databases for the distribution of these lipids. What other organisms can produce these lipids? We can do phylogenetic analysis to look at the evolutionary relationship of these biosynthetic pathways between different groups, different taxa. Actually, we can also do some of the biochemistry involved in the biosynthesis of these molecules. Something that's come out of our group is that a lot of the proteins that we're identifying that are involved in the biosynthesis are actually novel hypothetical proteins that have no known function. And so it's been kind of interesting to try to characterize these biochemically and discover what motifs they're using and what mechanisms they're using to make these lipids. Construction of deletion mutants also allows us to do physiological studies, so we can make a mutant that no longer makes this lipid and then test what conditions that not viable or what phenotypes we observe in terms of disturbing the membranes and whatnot. And we can also link it to environmental conditions through regulation. We can look at the, using qPCR, we can look at the expression of these genes directly under different environmental conditions. And that, all of those experiments together are really going to help us kind of constrain these biomarkers and make these interpretations much more robust. Okay, so in my group, we've been focusing on a subset of biomarkers, and these are the cyclic triterpenoid lipids, and these include molecules like these pentacyclic hopanoids. These are primarily produced by bacteria. They're used as biomarkers in general for bacteria, but some of them that are modified are used for specific groups. They're produced by, we estimate about 10% of bacteria right now based on genomes, but it could be more. And they're really diverse in terms of their metabolisms and the environments that they're found in. We've also done some work on tetrahymena, which is very similar to the hopanoids, the main difference being this last ring being a 6-carbon ring versus a 5-carbon ring. And this is usually used as a biomarker for water stratification. I don't have time to get into that, but we've been doing some, typically these are found in ciliates, but we've been able to detect it in anaerobic methanotroph, and we've been doing some work on this organism. But what I want to focus on today are the sterols. And so sterols are also used as biomarkers. They're also preserved in the rock record. Sterols are for eukaryotic cells. They're essential for all eukaryotic cells. If an organism can't produce sterols, it must acquire them through their diet. There's a lot of different, very important roles for sterols in eukaryotic cells. And the most well-known is the role in membrane fluidity, maintaining rigidity of the membrane under high temperature, for example, or making the membranes more impermeable. But there's also roles in cell signaling. Sterols are precursors to steroid hormones, which are important in development and embryogenesis and all kinds of different important developmental aspects of eukaryotic life. And like I said, they're also preserved in the rock record, and they're typically used as biomarker for eukaryotes in general. So cholesterol, cholestain is found in the rock record, and there's also very specific ones. For example, this is an isopropyl cholestanol that is actually used as a biomarker for sponges. So these are also used as biomarkers for Steranes. So when I started to look at how sterols were being used as biomarkers and what their role were in terms of in the biomarker world, I was actually surprised when I started to read in the literature that actually bacteria do produce sterols. It's not strictly restricted to eukaryotes. And so this really caught my attention because I love bacteria. And what I found was really interesting that at the time when I first started this project, it was known that about 3 different groups of bacteria and a total of like 6 strains actually produce sterols. So not a lot at all. And so those groups were aerobic methanotrophs like Methylococcus capsulatus, myxobacteria, which are very interesting delta proteobacteria that form these very unique fruiting bodies. They've been studied extensively for the ability to form these fruiting bodies. It's kind of a developmental model. And then one plankton mite, Gemata, has been shown to produce sterols. Again, another weird bug. organism is actually thought to have like a membrane surrounding its DNA, kind of like a nuclear protonucleus. But the interesting thing is that not all methanotrophs produce sterols, and not all myxobacteria, and not all planktomyces. So it's just a select few strains within these groups as well. And so I just really became interested in general and sterols in these organisms. And I had three basic questions when I started to look at this. Do other bacteria produce sterols? Are these the only ones? These were all done by lipid analysis. But now that we have all this genomic data, can we look in genomes and try to decipher if other organisms can produce these sterols? Is the synthesis of sterols in bacteria the same as eukaryotes? Do they have the same proteins to do this reaction? Sterol synthesis in eukaryotes has been studied extensively. So we know a lot about sterol synthesis in eukaryotes, and is it the same in bacteria? And I think for me, the most exciting thing would be to understand what the functional roles of sterols in bacterial cells might be. Why are these bugs making them? And why do we only see them in these bugs? And so to address the first question, I decided to look at what we know about the biosynthesis of sterols. So we know that sterols come from squalene, which is converted to oxidosqualene and then cyclized to lanosterol. So these are the first two steps of sterol synthesis. So I decided to just look for oxidosqualene cyclase genes in the bacterial genomes. And when we did this, we found that there were a lot more organisms than just the three groups that we had been looking at. So let me orient you a little bit. These are all bacterial sequences, bacterial genomes that contain an OSC homolog. Here are the eukaryotes. Dictyostelium is a mold, so it's not a bacterium, it's a eukaryote, but the rest are eukaryotes. The colors represent the taxonomy shown here on the right. And so we see myxobacteria, we see aerobic methanotrophs, which we saw before, and then we see Gemata, the planktomyces. But now we also see some Bacteroidetes, an alpha proteobacteria, a Microbium, and some cyanobacteria. And so the interesting thing is, of course, you can tell the taxonomy is not congruent with 16S taxonomy. So it does seem to have this probably occurred through some kind of horizontal gene transfer and a complex evolutionary history, which I don't think we have enough sequences to decipher in any way. Just we know that it's kind of complex. But the thing that struck me was that there are more organisms than we had anticipated. So then I went and looked at metagenomes. And so the next tree I'm going to show you is this same tree. All of the bacterial sequences will be in black, and all the metagenomes will be in blue. So when we look at the metagenomes, so here you see there's a lot more metagenome sequences that are going to be bacterial OSC sequences. And so the potential to produce sterols in bacteria is probably more broad than we thought, because we haven't actually been able to culture many of these organisms. I've done this analysis subsequently. I made this tree maybe 6 months ago, and the tree has grown a lot now that I've looked at other metagenomes. And so the potential to produce sterols in bacteria is probably more widespread than we are anticipating based just on genomes alone. So that's really exciting, I think, and especially thinking about what the role might be in these organisms and how that might be related to their environments. Okay, so finding oxidosqualene cyclase in a genome is a good indicator of sterol production, but it's not actually showing sterol production. And so we wanted to take this one step further and actually try and culture some of these organisms for which we have a genome and see if we can actually test the different types of sterols they're making. And so we took on this project. We were able to get all of the organisms listed on this slide. This is Jeremy Wei, my lab manager, and Xinxi Yin, master's graduate student. He graduated last year. And what they did was they cultured a whole bunch of these organisms and tested them for sterol lipids. And we had two goals with this. One, we wanted to demonstrate that, yes, they're producing sterols. And two, we wanted to look at what the diversity of sterols they were. Did they all make the same sterols, or did bacteria make different sterols? Were there different modifications on these molecules? And we got a lot of interesting stories that came out of this survey. And I'm going to focus on two things. I'm going to show you a story that came out of looking at the methanotrophs, and then a story that came out of looking at this Bacteroidetes, Eudorea adriatica. Okay, but before I get into that, I kind of want to go over eukaryotic sterol biosynthesis very, very quickly. So there are three sterol pathways in eukaryotes. What I'm showing you here is how plants synthesize sterols. So they take oxidosqualene cyclase and then make the cycloartinol molecule, and then through subsequent modifications end up making stigmasterol. Vertebrates and fungi have a separate pathway. They cyclize oxidosqualene to lanosterol, and then through a series of demethylations and reductions end up producing zymosterol. This is where we then see a split where fungi take zymosterol to produce ergosterol, and invertebrates take zymosterol and produce cholesterol. Okay, and so for the most part of all the bacterial organisms we looked at did not have pathways that were this complicated. Most of them actually either stopped at lanosterol or cyclartenol, so they had one step to make sterols. But some of the organisms did modify the sterols a bit further, and some of those included the methanotrophs. So what we saw with the methanotrophs is that they were able to take acetylsqualene all the way to what we call a 4-methylsterol. So they were able to demethylate at the C4 position one time. This is significant because if we look at what we know about sterol synthesis in eukaryotes, we know that C4 demethylation, so completely removing these C4 methyl groups, is actually essential in eukaryotes. If you block those genes, they cannot use the accumulated product, which contains methyl groups at the C4 position. Also, there's a role for this accumulation of 4-methylsterols in fission yeast as a signaling as a signal for low oxygen and cellular stress, which induces a whole bunch of transcription of stress responses in these fission yeast when they start to accumulate these 4-methylsterols. And so we were interested in why Methylcoccus capsularis not only produce sterols, but specifically produce these 4-methylsterols. And so we thought what we could do is we could knock out the genes that were responsible for demethylating at the C4 position and try to do some characterization of phenotypes, see if they were related somehow to low oxygen or some other kind of cellular stress. The problem was when we went into the genome of Methylcoccus capsularis texas, our lab strain, we found that it was actually missing some of the genes in this pathway. It didn't have the C14 reduction gene, but then it didn't have the C4 demethylation gene. So we had nothing to knock out to do our phenotypic analysis with. And so the first thing we wanted to determine was how are these methanotrophs actually demethylating at the C4 position? And we got lucky in a sense because one of the methanotrophs we tested, methylmicrobium acylophilum, didn't actually do the step. It actually stopped here. So it contains 2 methyl groups at the C4 position. And that was lucky because it allows us to do a phylogenetic analysis, a comparative genomics approach with a very closely related strain. And so what we decided to do to try to find these genes is we went into the JGI phylogenetic profiler and asked it to find all the genes in our organism, which is Methylcoccus capsularis texas, that are also found in these other organisms that we have tested and shown produce 4-dimethylsterols. So they carry out this reaction. But are not found in this organism that doesn't, and these other organisms that we've shown don't produce any sterols. And when we ran that analysis, we actually got lucky in that we only got 20 candidates. Typically, you do something like this and you get like 600 genes, which is completely useless. But with 20 candidates, that's a very manageable number. So I started to do some just genome gazing and looking at these different genes that popped up, and I kept getting this arborane, and I was intrigued by this arborane because it contained this dioxygenase and this reductase, but it also contained a transporter that had a sterol sensing domain. But other than that, there wasn't a lot of genomic context. But when I looked at some of the other methanotrophs, I found that this arborane here, which is similar to this one, was actually located right next to a bunch of the cyclase sterol biosynthesis genes. And so that gave me confidence that potentially these 2 proteins, or maybe this whole arborane, could be involved in that biosynthesis. And so Amy Banta and Yirang Cho, so Amy Banta is a staff scientist in my lab, and Yirang Cho is another master's student that just graduated, picked up this project. And so what we wanted to do was we wanted to delete this 1016, 1017, this dioxygenase and this reductase, and see if it would prevent the demethylation. And so one of the things about working not in E. coli or not in Bacillus is that you don't always have the genetic system. So Amy Banta was actually the one who was able to get us to make deletions in Texas. So that was her role in this project. And then she helped Yuang actually make this mutation. And so what I'm showing you here is a chromatogram of the lipids of the wild type strain, showing that it makes both the dimethyl and the monomethyl. And then when they knock out those genes, you see that it now only makes the dimethyl. So it looks like these proteins are definitely involved in the demethylation, which is very exciting for us. But we were wondering if potentially there were other proteins involved in this demethylation. And the reason we were wondering that is because if you look at the mechanism in yeast, to demethylate actually requires 3 proteins, right? And we only have 2. And so there was the potential that there was yet other proteins that were necessary for this function. And to get at that, what Eudoraenol did was she took this methylomicrobial organism that only makes these dimethyls, and she decided to put the genes from this other organism in there to see if she could get it to remove that methyl group. So here's a chromatograph for the wild type showing making dimethyls. Eudoraenol then went ahead and integrated both genes individually onto the chromosome, and you can see we are still only making the dimethyls. But when she integrated both of them at the same time, now we see that the dimethyls in this organism are being converted to monomethyl. So this is great genetic evidence that potentially these are the only two proteins that are involved. So what is the mechanism? So I've presented this at a couple of conferences and talked to a few chemists about it, and they have told me that these dioxygenases typically come with a reductase that is its partner, and the reductase doesn't actually function in the actual reaction, but what it does is it is responsible for re-reducing the dioxygenase so that the dioxygenase can carry out this reaction again. So our mechanism now is that this is actually carried out by one protein, this dioxygenase, which is then re-reduced by the reductase. And when I presented this to a couple of biochemists, they were really perplexed because it seems very unlikely that this reaction would happen in one step. So this seems like it could potentially be a novel mechanism. And so we're working on trying to do some in vitro biochemistry to determine if this is actually what's happening. The other thing we were interested in is to understand how broadly these proteins were distributed in genomes. And so we blasted these against the databases. And what we found was it actually has a very limited distribution. So I'm showing you a tree here of the oxidosqualene cyclases that I showed you previously. And in red is everything that has these demethylase genes that we just identified. And these are the only organisms that have this demethylase gene. So it's a very limited distribution, not only to just bacteria, but bacteria that specifically have this oxidosqualene cyclase and are potential sterol producers. So given that this looks like it's a novel mechanism for demethylating and that it's very restricted to bacteria, I think it's justified for us to say that we've actually identified a fourth sterol biosynthetic pathway. We have the three pathways that we see in plants and a fourth one that we see in bacterial species. And so that was really exciting for us. And so where do we go from here? So as I said, we think it's this one protein alone that is actually doing it. We need to verify that further through biochemical studies, and we're working on expressing these in E. coli for some in vitro assays, and we're going to pursue that. And we're also interested in what role these 4-methylsterols play in microbial stress responses. Now that we have a mutant that cannot demethylate at that position, we're wondering if that mutant will have some kind of phenotype in either some kind of stress, hypoxia, as we see in vision yeast, or nutrition starvation. The other thing is, now that we have this mutant, we have a strain that typically produces 4-methylsterols now being continually exposed to 4,4-methylsterols. And so we're wondering what kind of global responses that will elicit. If this is truly some kind of response to a stress, how does that change the transcription profile of the organism, and how does that change its protein profile as well? So we're kind of looking into potentially running some of these whole genome analyses to see how this is affecting the overall mechanism in the organism. Okay, so that's all I have to say about sterols. I kind of want to go off on a tangent now and talk a little bit about Eudora adriatica, the second story that came out of this sterol pathway survey that we did. And so Jeremy was responsible for testing this organism for sterol production. So Eudora adriatica is a very interesting organism. It's an marine organism, which is, from the rock record perspective and the biomarker perspective, is very applicable. A lot of the rocks that geologists look at are from shallow marine environments. It's part of the Bacteroidetes phylum, which is a gram-negative, which includes a lot of gram-negative bacteria. The most well-studied are the Bacteroides. These are gut microbiomes that have been studied for a long time, but there are several other classes within this phylum, and they're found all over in all kinds of different environments. And what really intrigued us the most was that no Bacteroidetes has ever been shown to produce these cyclic lipids, or reported to produce these cyclic lipids. So Jeremy went ahead and tested this organism, and he came to me. It was really funny. He came to me and said, "Yeah, it's producing some lanosterol. I see sterols." And so I've had to blow up. So this is the chromatogram showing here. I had to blow it up for you 100 times so you can actually see the lanosterol that it's producing, and this is the spectra for it. But it was being swamped out by these two other compounds, and you know the spectra for these. This end looks very similar to lanosterol, but the rest of it was not. similar at all. So Jeremy brought these to me. He's like, I don't know what these things are. And at the time, my postdoc advisor, Roger Summons, was actually doing a sabbatical with us. And Roger's a geochemist, and he actually is a walking spectral library. So you can show him a spectra, and he will tell you what it is. And we showed him this, and he was stumped. He was like, I don't know what these things are. So that was exciting and confusing and, you know, annoying because we didn't know what we were going to do. But what Roger did is he got us in contact with an NMR specialist who actually works on plant molecules, plant triterpenoid molecules, like these cyclic molecules that we study. And and that was Jose Hiner at State University of New York in Syracuse. And Jose was actually able to identify these molecules. They look very similar to what some plants are producing. They look similar to our hopanoids, but they're being generated from oxidosqualene. These are completely novel. These are new molecules that have never been seen in nature before. So we got to name them. We're naming them Eudoraenol and Adriatical. And Jose was very excited about it because this is the first new identified triterpenoids in 30 years. And so that was exciting. And most of these are actually found in plants. So this is what plants tend to produce. Plants produce, in addition to producing these tetracyclic molecules, they produce molecules like isoorbanol and things like amarin and lupiol. And they have a whole bunch of functions for these molecules. A lot of them are important in defense. A lot of them, they don't know what the function is. But these are also preserved in the rock record. You can find these structures, and they're actually used as plant biomarkers, or they're used as indicators of terrestrial input into aquatic environments, and particularly isoorbanol. And so it was exciting for us because this molecule is actually used as an indicator for flowering plants, and we seem to have a similar structure here. And this is significant for the rock record because this isoorbanol, which is, again, a biomarker for flowering plants, has actually been found in rocks that are as old as the Permian and the Triassic. And that's significant because if we look at when flowering plants first appeared, it seems that they appeared in the Cretaceous. And so you have that are attributed to flowering plants actually occurring in the Permian and Triassic, which is 100 million years before that. And so what has been proposed for many years is that there was a microbial source, and this was kind of an orphan, what we call arborane biomarker. And so we're excited because our molecule might actually, you know, our organism might actually be producing and might be considered a source for this orphan biomarker. The other reason this is exciting is because this would be a new bacterial cyclase. And these enzymes are really interesting and have been studied extensively. So we know that there is a bacterial cyclase to make hopanoids. There is a bacterial cyclase to make sterols, and that's the cyclases that we have been studying. And now this will present a new class of cyclases that actually generate this pentacyclic molecule, these what we call arborinols, and this would be an arborinol synthase. But the problem was we had to verify this because it was possible, because of the small production of lanosterol, that this cyclase was actually not producing Eudoraenol, but rather we could envision that the pathway went this way. So that instead of, it was a normal typical lanosterol synthase, but then there were subsequent proteins that were actually catalyzing this conversion of lanosterol to Eudoraenol. Bacteria are tricky, and they will do things like this. So you have to kind of be careful and test this. And so what we did was Amy got involved in the project. And what she did was she set up a system in E. coli where she was able to place on a plasmid the gene so that from methanotrope so that E. coli could produce oxidosqualene. And then on a second plasmid, she introduced the Eudorea cyclase and saw that in E. coli, it will actually produce these two molecules that we observed in a native organism. So this enzyme is definitely producing these molecules. So it is a novel new bacterial cyclase. The other interesting thing we thought and potentially thought would be a problem is that this isn't actually a bacterial enzyme, but rather it's a bacterial enzyme that was acquired from plants. Again, these plants are known to produce a lot of these different molecules, and they have a very similar molecule in isoorbanol. And so what we did was to try to determine if this was related to the plant cyclases. We reconstructed our oxidosqualene cyclase tree, but this time included many more of the plant cyclases and our Eudorea cyclases. And that's this tree you see here. And so what ended up happening is, this is the Eudoraenol cyclase shown here, clustering by itself next to all of the bacterial oxidosqualene cyclase. And here are the plant cyclases. And within this clade is the eukaryotic, the plant isoarborinol synthase. So clearly this R cyclase is not related to what we see in the plant cyclases. So this was exciting. We have a new molecule. It's potentially helping us identify a microbial source for this orphan biomarker, and it's a new cyclase. What we next wanted to do was, can we distinguish between this type of a cyclase, an arborinol synthase, and a sterol cyclase in the genomes and metagenomes? Because if you remember, I pulled this Eudoraenol cyclase out by doing a search for oxidosqualene cyclases that made sterols, and I thought it was a sterol synthase. So is there a way at the amino acid level that we can start to distinguish between these by just sequence alone? And so the first thing we did was we did an alignment of the Eudoraenol cyclase with other sterol cyclases. And I'm showing you here just a truncated cyclase. So this is the human oxidocyclase. This is the sterol cyclase from methanotroph, and this is our new Eudorea cyclase. And I've highlighted a few of the residues that we know are important in sterol synthesis, in cyclizing oxidosqualene to sterol. And in particular, what stood out was that in Eudorea, this histidine was converted to a tyrosine, and this tyrosine was converted to a valine. And they're very far apart from each other on the primary amino acid sequence, but if you look at a model of the human oxidosqualene cyclase, which we have a 3D model for, these two residues Amy has highlighted here in red, and this is the sterol molecule. Even though they're far apart each other on the primary amino acid sequence, they're actually very close to each other in the structure, and they're both in the active site, and they're both right next to where the fifth ring of Eudoraenol would be generated. So it seemed like these were very important. And so maybe these could be diagnostic for a cyclase that is actually making arborinol instead of a sterol. And so the first thing we thought was, well, can we actually show this? Can we convert these two back into what we see here? Oops, my phone is reading. Oh, it hung up. Can we convert these back to what we see here and get this to actually convert back to being a lanosterol synthase? And so Amy did some site-directed mutagenesis. And so here's the wild-type protein being expressed in E. coli, and you can see the two peaks that we would expect. She makes the substitutions, and nothing. We lose the enzyme. So it looks like these might be important, but we aren't able to convert it into a lanosterol synthase. And so we thought, okay, well, let's try it the other way around. Can we then take a sterol synthase and then change these to what we see in Eudorea and convert it to make the pentacyclic molecule? And when we do this, we actually start to see a little bit of the molecule being produced. So here's the wild type. It makes, in E. coli, it makes a ton of lanosterol. It's going crazy. It still makes a lot of lanosterol when we make these substitutions, but then you start to see a small amount of one of these molecules, which we call adriatical. Okay, so it looks like, yes, these potentially could be playing a role, and they're important, but there's probably other amino acids that we could look for. And so Amy just did some basically gazing at the active site, and she pulled out these two other residues that were changed in Eudorea. So this one, 230, is right next to the histidine, and it goes from being a very big tryptophan to a smaller serine. And then this asparagine further down is actually here, and it's changed to a much more bulkier molecule in Eudorea. So these are conserved in all of the Eudorea cyclases that we could find in genomes that we think are Eudorea cyclases. And so Amy went ahead and constructed 4 substitutions in the sterol molecule, changing them all to these yellow ones. I'm sorry, in the sterol protein. And when she does that, when she makes those 4 changes in a sterol cyclase, we now get to see that it is actually making a variety of these pentacyclic structures. So we still make lanosterol. This is all in E. coli, so the protein's being expressed in E. coli. Still makes lanosterol, still makes Parkial, but now we see our adriatical, which is from our organism. We see isoarborinol, which is the one that we had been observing in plants, and we see a little bit of Eudoraenol, which is the one that Eudorea adriatica is making. So what we have here are 4 diagnostic amino acids that we can look at. And so when we search genomes and metagenomes, we actually find that those that are most closely related to Eudorea have these 4 changes. And these are outlined here. And it's interesting, the only cultured genome organism that has these changes in Eudorea. So this is probably the only cultured organism that makes these types of molecules. And it's funny because we probably never would have found it if we didn't have the genomic sequence to guide us to that potentially this was making some kind of cyclic molecule. And then we have two different clades in terms of the environments that they're finding. We have this as being the marine clade, and we also have some potential sources in freshwater environments, but there's no cultured organisms that go with these. But I think what it's showing is that there are potentially other sources for these biomarkers out in the environment that are microbial. And so what we want to do now is we actually want to have these synthesized, and we're going to express them in our E. coli system and try to determine what they're making, whether they're making these pentacyclic molecules as well. And we think this is a great way to try to identify other novel cyclases. Can we look at amino acid sequences of all those metagenome OSCs that I highlighted to you? Can we see significant amino acid changes, express them in E. coli, and then see if they're making a novel molecule? And of course, we want to study the physiological effects of these lipids. What are these lipids being used for? It's great that we discover the proteins to make them and that we discover the organisms that have them, but ultimately, I'm really interested in their function and what their role is in these cells. Okay, and so with that, I hope I've convinced you that we can utilize these molecular techniques that we've used to kind of inform us about important aspects of biomarkers. You know, in the first example I gave you, I showed you a novel pathway for a biomarker that is typically restricted to eukaryotes, but we see that bacteria also have these biomarkers and they have unique ways of modifying it. And then I've shown you how we can use these tools to identify sources for what we call orphan biomarkers potentially, and we can use these to hone in how we find these biomarkers by looking at the amino acid sequences and really pinpointing where these molecules might be important. Okay, so with that, I would like to thank my group. Of course, Jeremy and Amy contributed significantly to all of the work here. Yuang did the work with the 4-methyl sterols, and also acknowledge our funding sources. And thank you, and I'd be happy to take any questions. Thanks.
Cool. Thanks much. Actually, I have a question to start off with. So my background isn't in any of this, so I'm wondering what the, like, why would there be different sterols in Eudorea? Like, what could the possible purpose of that be, and why modify them if it's not for signaling? You know, if it's for membrane fluidity, why modify them? Or if it's for signaling, do you have any evidence that that actually changes how these bacteria might function in nature?
Yeah, so that's the thing. Everyone says they're involved in the membrane, that there's this membrane function for them, and we don't, you know, we're not sure. You know, one, they don't make them in huge amounts like you see in eukaryotes, and so I find it hard that they would be modifying the membrane completely. So yeah, I think they're made in low amounts, and I think they would be signaling is what I think might be important, that they provide some signal, but we don't know. We just don't know why these guys make them. And the thing about Eudorea is it makes a lot of different lipids. It's lipid profiles, so it has a lot of these lipids, and it's unclear why they would make them. And you know, plants do this. Plants make tons of these molecules, and there's this idea, oh, they're important in defense. Some of them are toxic, and that's what they're used for. But and some plants make 40, 50 types of these molecules, and we're not sure what their functions are in there. So we're not sure what the function is in bacteria as well, but we're really, I mean, that's really the goal, to find out how these guys, what they're using them for, because I think ultimately that's really going to help us when we see them in the rock record to try and interpret them a little bit better.
All right, I'm going to look online. Jen, you have anything to add, or?
Paula, you might want to stop sharing so we can see you again.
Oh, okay.
Yeah. Okay. Sorry. So I have a question actually following up on your, there you are, hi. Following up on this idea that microbes can make what is thought to be only a flowering plant sterol.
Mm-hmm.
I mean, what does that mean
for the
ancient biomarker research?
Yeah.
And like how, I mean, hopanoids, like we knew that we had problems with hopanoids, right? But like this idea that you're now replacing flowering plant biomarkers gets even more testy, right? Like,
yeah, and so I mean, so this is something I'm not too familiar with, because it was one of these things where it was like, oh, I had no idea this was something people were looking for, and we found it. So really, all I've seen about the rock record are those few papers that found them in the Permian and Triassic. They actually find them in like anoxic environments. And so again, an idea that like there couldn't have been a plant present here. There had to have been some kind of microbe, right? And of course, this microbe is aerobic. You need oxygen to make these molecules, I think. So there's still potentially other sources out there. But I think, primarily from what I understand is these plant biomarkers are used in more, they're old environments, but not old in geological terms. They're like 100,000, 200,000 years ago as an input in like terrestrial input. You're seeing terrestrial input. And so now you kind of say, well, we have freshwater environment, freshwater ones that are not freshwater sources potentially. We don't have a bacterium, but it looks like we might have some. So maybe they're just already there and there is no input, right? And so that's, I think, something that we have to start to consider, that maybe it goes beyond just these flowering plants. But besides the studies that were done on the Permian and the Triassic, I'm not sure if there's other studies that have looked further back and seen them. I'm sure there are. I'm sure the oil people have tons of these somewhere in their samples.
Well, I was also wondering, that you see this great diversity in that one organism can be making, what was it, like 7 or 8 different types, right? But then if we think about like what is that going to look like in 100 years, would the breakdown products look similar for those, or would there be like residual? I mean, everything turns into the bitumen eventually, but like would there be residual bonds kind of left on the edges somewhere, or would they look different years from now, basically?
Yeah, they would, and I think they will look different. So that's a whole area of that I think I don't know if like Roger, I think, is trying to get back into that, because I think he feels like, we need to see how these things break down to understand what it is that is breaking down. And I'm not sure, I don't know anyone that's doing that kind of stuff. But I mean, that's how they found the ones from the Permian and Triassic. They're actually breakdown products of isoorbital. And where those bonds are, so the organism that we have is making something that looks like isoorbital, but not exactly isoorbital. The double bonds are a little different. And I don't know how that's going to change the chemistry of it breaking down. So it might look completely different than what you would see in the rock record for one versus the other. So, and those are more liable when they have that hydroxyl group at the end. I mean, there are ways that it can be broken down much more easily than, say, the hopanoids, which are all completely saturated, and those are hard to break. Yeah. Okay. Cool.
Well, that was super cool. Did we get any questions coming in? Some people are retweeting things, but I don't see any questions.
Okay.
I do have one, just it's in the back of my mind too, but I have no clue whether it's related to this or not, but I know in like choanoflagellates, the sterols modify, like they have this whole clumping behavior that the sterols are modified by, right? So I'm wondering if there's potentially, I don't know, there's this story of like bacteria in early eukaryotes kind of allowing for multicellular eukaryotes. So it kind of all in my head kind of comes together with that. But Nicole Keene has found that lipids are really important for a lot of the stuff in choanoflagellates. That's, I think, really exciting, because I think, looking at how lipids affect and do these kinds of things and affect other organisms, is I think interesting. Something that we started to get into is to look at sponges, because we find that a lot of the sponge symbionts have these sterol biosynthesis genes. And they have a lot of these modifying genes. Actually, these demethylase genes are found in symbionts of sponges. And so that's really cool, because then it's like, oh, well, who's making the sterol? And is it the sponge or is it the symbiont? And how is that affecting it? And why? Why would it need to modify it? What purpose does it serve in a sponge, which can get its sterols from its diet? And so there's a lot of like how these lipids affect other organisms, particularly eukaryotes, would be really interesting.
Cool. All right. Well, I don't see any other questions coming in. So thanks so much for doing this, Paula. It was an awesome talk.
No problem. I had a good time.
All right. And thanks, Dave, for setting it up.
Yes, thank you. All right. You guys have a good weekend.